Can anybody tell me what the main organs of the urinary system are? Kidneys. I like that answer. Kidneys. And what do the kidneys do? That's so generic. We're way past that now. I don't know. You want to say filter. They filter. What do they filter? They filter the blood. Very good. And when they filter the blood, what does that really mean? Clean, pulling out water and junk, waste. I heard all of y'all. Here's what we want to know. Yes, it does everything that I just said. But there are two ways to think about what the kidneys do. If they filter the blood, we want to look at it two ways. And the first way is the way that everybody looks at it. The kidneys make urine. They take the bad stuff, the stuff we don't need, out of the blood, right? And they turn it into urine. That's the simple understanding. They filter the waste. It's actually deeper than that. And realistically, what I like to say is the kidneys determine what your body needs, and they allow that to stay in the blood. That's a real fine distinction. It's like looking at a cup half empty instead of half full. See, if we look at it half empty, we would say maybe think about what it's taken away. But we want to also be able to say the kidneys filter the blood, and when we say filter, they leave the good stuff in the blood. They leave the things in the amounts that we can utilize in the blood. And here's a little slide that talks about that. Remove certain stuff from the blood. But really, what is it doing? It's regulating the chemical composition of the blood. It's maintaining, could you say it's maintaining homeostasis? Yeah. Maintaining homeostasis of the composition of blood. But we also know from what we've talked about before, and I'll say two words, renin-angiotensin. You guys remember that? I don't expect that you remember the whole thing perfectly, but the kidneys actually play a role in monitoring and regulating blood pressure, blood volume and blood pressure. And they do also determine the pH, and this won't make sense necessarily yet. Well, this part will, and we just don't have a name on it. What if your kidneys decide to get rid of more hydrogen ions? Does that change your composition of blood? Sure. Does that change the pH of blood? Sure. What if your kidneys decide to hold on to more hydrogen ions? Does that change it? Yeah, so we can see it regulates the pH by that. That's pretty cool. And the only one that's not on here, which I guess I'm going to add right here, is the kidneys play a role in activating vitamin D. Oh, but it says blood volume, and we know that blood volume goes right along with blood pressure. Here's the kidneys. We know they're up here. We know these are called ureters. There's a bladder. What's the little tube here? Urethra. How many urethras do you have? Proper answer hopefully is one. I don't know. Some of y'all may have two. If you do, keep it to yourself. We don't need to know. When we get serious about the kidney, we're going to be talking a lot about its anatomy. Well, I mean, you're going to be listening a lot and learning a lot, and hopefully you learned a lot about the anatomy. Because you need to understand that this outer part of the kidney is called the cortex, and this inner portion here is the medulla. However, the book does not do a good job here because they say renal medulla right there and point to this. This structure is not called the medulla. This structure is called a renal pyramid. So is this one and this one and this one. And this one and this one and this one. Want to tell me why they're called renal pyramids? Because they're shaped like a pyramid. That's exactly why. They happen to be located in the medullary portion of this organ. If the outer part here is the cortex, then this is the medullary region. That's the difference. If I say name the structure, it's a pyramid. If I say name this area or region of the kidney, then it would be the medulla. And I'm not real big on renal medulla. I'm huge on renal cortex. But renal medulla, really, we call these things the pyramids, and that's what we'll be talking about. Why do you see red and blue here? Blood vessels. If we're going to filter blood, we better bring blood in. Who agrees? Good, 3 people. I like that. What kind of blood would go into the kidney? Arterial or venous? Leaving the heart, going through the body. Part of going through the body is going to the kidneys. Arterial. Absolutely right. So the renal artery is where it comes in. What artery was it in before it was in the renal artery? Very good. The abdominal aorta has a branch called the renal artery. You might remember it's the 3rd branch off of the abdominal aorta. We kind of talked about that a lot in lab earlier this semester. And then the renal artery comes in and it branches and becomes the segmental and interlobar and the arcuate here, and then it gets real tiny out in the cortex. My point right now is not to get you to write all those down. That's what the lab integrity is for. Look, here's a slide right under it that gives you the entire blood flow all the way into the kidney and out of the kidney. Once again, my focus right now is not to get you to do that. There's plenty of good stuff where you can figure that out on your own in the lab talk. My point is to get you to see this. Blood comes in and moves out to this area of the cortex. Most of the work of the kidney is done out there in the cortex. Okay? The filtering is done out there. And it's done in these little groups called, or these little units called, anybody? Nephrons. You have about 1.25 million nephrons per kidney. Most of them are out in the cortex. You do not see them on this picture because they are microscopically small. Okay, so let me make sure you understand. Blood comes in, moves all the way out here to the cortex, then it gets filtered, right? So at that point, we now have blood that's going to need to pass back out, but what other fluid will also be produced that needs to pass back out? Urine. At this point, we're being very generic about that fluid. Technically, I'll show you in a minute where we don't call it urine yet, but that's for later on. So we have one fluid coming in, blood. We have 2 fluids coming out. We have both blood and urine. Well, we're being real generic when I say the kidney filters right there. There's actually 3 processes, and we're going to go through all 3 processes. Blood in. Notice it goes through the vessels. They kind of go either up there, maybe come through here, gets out here, and this is where the nephron's located. That's where the material gets changed between the blood and the kidney tubules. The blood goes back through the blue blood vessels, which we might call veins. And where do you think the urine goes? You see this pyramid? It comes right through these pyramids. You see those lines there? Those are big tubes that we're going to talk about in a minute called collecting ducts. I'll show them to you. And then right here, the pyramid opens up into this open space. They call these calyces. Then it gets a real big open space here. It's called a renal pelvis. And now it's in the ureter. And it can travel down to the bladder. Kind of, once the material is in the collecting duct here, we can start to refer to it as urine. Oh, and this, right now you go, why do you say collecting duct? That's a pyramid. Because that's what the stripes are, and when we see the nephron, you'll see that in a minute. So once it's in the pyramid, we usually can typically refer to it as urine at that point in time. So I guess really what I need to do is show you a nephron. Notice, we always orient ourselves to pictures. They're taking a little chunk out of the border right where the cortex and the medulla come together here, or we could say cortex and a pyramid. So right here, down below, that would be a pyramid. They're not showing you the whole pyramid. Okay? Up here is the cortex. Now they've done something a little odd here. They're showing this loop of Henle all the way down in the medulla. It's not always like that. It can be more or less. So what we'll generically say is this, that the nephron is typically located in the cortex. Even though it may have little areas that go into the medulla, it's typically considered cortical. Now we've already said we push blood through the kidney so that blood can eventually be filtered. Look right here. This is the location where the blood flow and the tubules finally meet. We kept blood separate and segregated all the way through here. You don't see all the vessels in this picture, but I'll tell you, they start here with the renal artery. It goes segmental. It goes interlobar. There's one that arcs over the top of the pyramid right here. That's what you see here, called the arcuate. Then it radiates outward from that into the cortex. They would call this cortical radiate. You see it named on another picture. Then we have flow going into and out of right here. What color are these? Red. And they're the tiniest little arteries possible, so technically they call these arterioles. This would be your afferent arteriole, and this would be your efferent arteriole. And what they go into and out of is this ball of capillaries right here called the glomerulus. Now what they are showing us is they're actually showing us the Bowman's capsule surrounding the glomerulus. And that forms a unit, this entire thing here, they like to call a renal corpuscle. Renal corpuscle consists of the glomerulus inside and the Bowman's capsule around it. And, you know, they've tried to do 3 dimensions well, but I wish they would have just drawn this like it was sliced open so we could see it. Because it's a little hard on the screen up there to see this area. It looks like a little chunk taken out. But they're just trying to make it 3-dimensional. Anyway, what happens here? If these are capillaries, what occurs? Good answer, exchange. Right? Except now we're changing it. I know we've always said exchange, but now we're going to say that right there in this part of the kidney, it's not really two-way exchange. It is fluid and lots of other stuff, whatever's in that fluid, moving out of the capillaries and getting trapped by this membrane. Man, when you see that like that, you're going, holy moly, how much is there? I don't expect you to be able to draw it all at once. I'll come back and draw this for you in a minute. But first, I want you just to look here. This is a ball of capillaries. Oh, and I know, I forgot. That little bitty thing doesn't show up. Right here, when we're looking here, this is the glomerulus. It is a capillary bed. So all these are capillaries in here. The Bowman's capsule surrounds it, as you can see going around right here. And going around right there. That's the Bowman's capsule. Comes all the way around here to where it becomes the proximal convoluted tubule. What takes place right here is simply filtration. That is movement from the capillary outward. That's one-way movement. Anybody want to take a wild guess why the movement's that way? Why it's pushing out? Because what takes place here, and notice I put the 1 right there and circled it. It's because we're saying right here the movement is all one direction, and it's movement due to pressure, from high to low pressure. And we call that, in AMP1, we call that filtration. So the first part of the formation of urine is this. When the fluid gets into the glomerulus, it gets pushed across due to the pressure. And we call that filtration. Then it gets trapped by the Bowman's capsule. Then the fluid has no choice but to continue through the tubules right here. First we have a proximal, that means first. And it's curved, so they call it convoluted. Eventually it takes a dip down, they call it the loop of Henle. Comes back up over here and starts to curve again. They call it the distal convoluted tubule. And then that joins a larger vessel right here called a collecting duct. You can see that many other nephrons, many other distal tubules are actually joining this collecting duct as well. So collecting ducts are larger structures in which lots of nephrons can dump their fluid. So by the time the fluid comes into here, we can start to refer to it as urine. When the fluid's out here in these tubes, and I don't care if it's here, or out here, or in here, we still refer to that fluid as filtrate, usually. Commonly, we even call it glomerular filtrate. I'll say it now, but I'll say it again later too. These are the tubules, the kidney tubules. You saw them in AMP1. They're simple cuboidal epithelium. We even saw them in this lab last week. Simple cuboidal tubes, most of them. There's a little part of the loop of Henle that's not cuboidal. For our purposes right now, we're not worried about that or concerned with that. However, here, this is what? This object right here. That's the glomerulus, so it is a capillary bed. It is simple squamous epithelium. Also, the Bowman's capsule that surrounds it here is also simple squamous epithelium. The PCT is proximal convoluted tubule. DCT is distal convoluted tubule. What does proximal mean? You guys remember? Close to. Okay, this is the main worker of the kidney. This is the glomerulus. This is where fluid and lots of other stuff initially go from being in the blood to getting their way into the tubules. What pushes them over? Pressure pushes them over. So it moves out. We call it glomerular filtrate. That's what this little GF is. You can just add it to the drawing that you already have, maybe. Or you can label it in a picture somewhere as well. Now I'll show you what was missing in the other picture. In the other picture we had this glomerulus. We had this Bowman's capsule. Do you remember what we called this little blood vessel? Afferent arteriole. What about this one going out right here? Efferent arteriole. Very good, guys. So these are connected to other vessels. I'm just not showing them to you here because blood comes up and in here. And then whatever does not get filtered continues on through here. This tubule network houses the filtrate, and it curves all along. And whatever eventually ends up into the collecting duct right here, we could finally call it what? We could call it urine. Here's where I want you to get a little bit more. This tubule right here, as soon as this fluid enters the proximal tubule here, right away it starts to move out of the tubule and back into the blood. Okay, the tubules are permeable to water. They're permeable to solutes in most locations. We'll get real particular with that later on. And so instantly, material moves right back into the blood. We call this process, and you can see it hinted at up here, but down at the bottom it's called there's a 2 around it, and you see it's called reabsorption. Reabsorption is movement from the tubules back into the blood. Okay, so we're filtering, right? That's what the kidneys do. That's what we've always said. The kidneys filter. They filter the blood that comes into them. So now what you're doing is you're able to see the little tiny network called a nephron that does this job. Fluid moves into the glomerulus, and it is immediately filtered across. Here's the crazy thing. How many of these glomeruli do we have in each kidney? How do you know that? Because one in each nephron, right? That's the main part of the nephron. There's only one of them in each nephron. So we're saying that overall, you have 1.25 million of these in each kidney. If they could add all the fluid in one day that moves across here and gets captured by the Bowman's capsule, another way to say that is if they could calculate all the glomerular filtrate that is produced in one day across here, it would be this. 180 liters a day. That is crazy. 180 liters a day of glomerular filtrate is produced in an average person. That's if you total up all the 2.5 million nephrons that we have, all their glomeruli, 180 liters a day. Do you drink 180 liters of water? Then how do you filter 180 liters out a day? Well, it doesn't have to do with the oxygen. It just has to do with the fact that how many times a day is your blood circulated? You have 5 liters of blood, but it gets pumped through the kidneys all the time, right? Goes to the kidneys, goes to the kidneys, goes to the kidneys. When they add up how much of the cardiac output goes to the kidneys and how much filtration occurs in a day, that's the number they come up with. 180 liters. Let me ask you this very simple question. How much urine do you form in a day? 1.5 to 2 liters. Average person. Wow. If we're only making 1.5 to 2 liters of urine a day, but we're filtering 180 liters, where's the 178 go? Where's the other? It goes right back, excellent answer, right back into the blood through what process? Reabsorption. Now, all of that doesn't move right away at the proximal tubule. Some of it moves in the loop of Henle and some in the distal tubule, but well over half a day moves in the proximal tubule. It's a real active part of the tubule. But even then, once we take the stuff out, this we still call filtrate. Because even at that point, we're not done fine-tuning. There's one last process. Look right here. Sometimes some things get reabsorbed into the blood that the body then determines, wait a second, we want to get rid of them. So there's a third process right here. The third process is called secretion. From the blood back into what? The tubules. Notice how I put secretion also up here. See how there's a little 3 here? That's telling you that secretion can take place in any of these areas. Look at this picture. Tell me some parts you can label already. Glomerulus, right? Everybody could label that. Look, they've turned it a different direction. Don't be freaked out by that. Okay? This is a glomerulus. So what do we call the tube, the blood vessel that leads into it? Afferent arterial. What do we call the blood vessel that leads out of it? Efferent arterial. Remember earlier when I showed you proximal tubule, loop of Henle, distal tubule? What they're doing is making it one little tube representative of all the kidney tubules. So they're not breaking this down between proximal, distal, and loop. This is a big picture concept to show you what are the 3 processes that lead to urine formation. Look, this is a great Bowman's capsule right here. They've cut into it. We're seeing how there's actual space, like you actually saw in lab, if you're taking it on campus, and you saw those pictures, you saw that there was like a lumen here, right? Where stuff could move out of the blood vessel, and it gets trapped in this lumen, and then it moves here. So what process takes place here, which is movement out of the capillaries and into the tubule for the first time? That is filtration. That is filtration. What I never got back to, but kept showing you and kind of talking about like you understood, is this. Around all of the tubules, there have to be capillaries in order for material to move out of the tubule back into the blood for reabsorption. Or for stuff to go from the blood back into the tubule, there have to be capillaries. So we didn't see it yet, but I'm about to show you a picture that will show you those capillaries. Okay, so just bear with me there. Then, go back to here. Blood moves in, gets filtered across. We now call it glomerular filtrate. How much a day? 180 liters. And by the time it passes out of here and becomes urine, how much? 1.5 to 2. So what happens to most of it? Process 2 right here, reabsorbed right back into the blood. Notice they call the blood vessels, the capillaries that surround the tubules, peritubular capillaries. That's their name. It's a great name. It means capillaries that surround the tubules. And once that has happened, we can also have a little bit of secretion. Keep in mind this third process that we call secretion, which is just fine-tuning, putting whatever back out that we don't want, right, taking it out of the blood. I want you to go ahead and write this down. There's only a little bit of stuff that's secreted back into the tubule. Mostly that would be hydrogen ions, potassium, certain medications, and that's really the big deal. For secretion, the big idea is hydrogen ions, potassium, and medication. So we're going to spend a lot of time talking about reabsorption. We won't talk about secretion much because it's the least of the three processes. And filtration is very easy. So let's see a real nephron again. Tiny. If we zoomed up, you could follow the renal corpuscle with its Bowman's and glomerulus. You could follow the proximal tubule out of that, the loop of Henle, and then the distal tubule. But I'm not zooming up on it that much. My purpose here is to show you that there are significant numbers of capillaries around the tubule network. Even on some of the special nephrons that dip really low into the medulla like this, they have their own special capillaries around their loop of Henle. Those special loop of Henle capillaries are called vasa recta, and they are very similar to the peritubular capillaries. That is discussed in the lab talk. Okay? It's just more capillaries. What is this big tube here? Collecting duct. And what part of the kidney is it found in, within? Good. This would be a pyramid-shaped object. They're not showing us that, and most of the slices in the pyramids would actually be these collecting ducts that are opened up. Let's draw another one, and let's talk about it as we draw it. We have blood move into the kidney. Okay, it goes through lots and lots of blood vessels. It finally gets to the cortex where we start the work. And it enters the glomerulus through a little structure that we could call a what arteriole? Good, an afferent arteriole. So I'm just going to make this bottom one the afferent arteriole. No, I don't care how perfect this looks. The point isn't to make it look perfect. The point is to get you to understand that these are a bunch of capillaries. And when blood is in here, is it under some pressure? Absolutely. And that pressure will push the blood and lots of the material that's in there straight across. And yet we don't want that to just ooze through a kidney. We want to collect it, trap it, right? Send it through a special network. So here's what we do. Put a Bowman's capsule around that glomerulus that will collect that fluid. Now, you will see in the lab talk that this is the parietal, the outer layer, and there is actually part of the Bowman's capsule that sits right on these capillaries here. And if this outer part would be called parietal, take a wild guess what we would call the inner part. Visceral. But that's not my focus right now. What do we call the fluid that continues this way? Pardon? Very good. We call it blood. Because what's the name of the vessel that we're within? Efferent arteriole. So what's in an artery and a vein? Blood, right? This is an arteriole, so it's blood. Just, making sure that you know what we're talking about here. Those efferent arterioles would lead to a bunch of capillaries. I could continue them on from there, but I'm just going to show you, like we'll put one here. They're continuous. This would be a peritubular capillary. Notice it's surrounding the proximal convoluted tubule. Why are they calling it convoluted? It's twisted, it's turned. Yes, that's what convoluted means, twisted, tortuous, curved. So it does that. It kind of loops around and curves around and has its various little shape to it. But then eventually it always does this. It will always dip down at some point like this. And this little dip right here is called the what? Loop of Henle. You do need to absolutely be able to trace that this would be the descending limb because fluid is moving down, and this is the ascending limb because fluid is moving back up. Now the way that it works is this distal convoluted tubule typically, I'm going to erase just to make it go where I want it to go. It typically comes back around, and sometimes even before this book has changed it, and they now say the loop of Henle does this. But it goes back and it touches the afferent arteriole, or gets really close to. It actually does come in contact with the afferent arteriole. Then over here we can put a big old collecting duct that this tube joins. So what is this tube right here? That's the distal tubule. What is this here? Collecting duct. Those are the parts of the nephron that we see here. What is this process that occurs right here called? Filtration. What is the process that is movement from the tubules, no matter where? Look, I can put capillaries all over the place and point that direction. We call it, when it moves this way, we call it reabsorption. So that's process 2. So we have filtration, reabsorption, and then finally secretion. Now, secretion can take place. It likes to take place in the distal tubule, but hey, it can also take place here. If we move it this way, back that away, then it's secretion. 3 steps. From the blood to the tubules, only at the glomerulus and Bowman's capsule, that's filtration. Anywhere along the network, from the tubule back out into the peritubular blood, we call that reabsorption. And then from that blood, if it goes back in to the tubule, we call it secretion. And that could be anywhere along there as well. Lots of it's at the end. That doesn't mean it has to be at the end. How much glomerular filtrate is produced a day? 180 liters goes there. How much urine? 1.5 to 2 liters a day ends up here. That means that most of it is reabsorbed somewhere along the chain. Next, very important concept. So we're talking about the filtration that happens right here at the Bowman's capsule. What leaves the blood there? What actually leaves the blood? We know that everything, not all of the fluid can leave because we still have blood here, right? But what does leave? Here's what's important to know, that by the time the blood goes through your kidneys, the blood that's leaving your kidneys has virtually no sugar in it. I said the blood. Oh, Dr. Cooper, you messed up. By the time the blood leaves the kidneys, the blood has recaptured all of the sugar. What I mean is your urine has no sugar. But here's the cool thing. What I really want to talk about first before I even get to that is right here at the glomerulus. What moves across? Everything except what is too big to move will move across there. And the things that are too big to move would be blood cells that are in there. Those are too big to move, and protein. Virtually everything else is filtered out and makes it into the tubule. Okay. What is this? Bowman's capsule. See how you're getting used to seeing it? So what does that make this? Proximal convoluted tubule. You'll notice we commonly say just proximal tubule. You need to know the word convoluted. That means it's curved, but they leave it off a lot too. You just need to understand it. What would be surrounding this tubule? Some what? Some capillaries. They're not showing it to you though, but we understand. What's in here? Well, what is this? What is this whole thing? Okay, that is a glomerulus. Therefore, that makes it a capillary bed. That means it's formed from simple squamous epithelium, and inside of those capillaries is blood. Part of that blood is red blood cell, correct? Look, they're showing you red blood cells are big. They're also showing you that this Bowman's capsule in this picture, they're actually showing us, and I think you see it better even if I do that. They're showing us that Bowman's capsule not only is on the outside, but it comes in contact. See this right here, how it's yellow? They're showing you that it actually contacts all of the capillaries. If you look up here for a second, sorry I don't have a webcam, you can't see it if you're at home, but they interlock. That visceral layer, it interlocks around the capillaries like my finger's interlocking here. Look right there. See how that happens? So what's going to squeeze out is only what's small enough to squeeze between these little spaces. Can you obviously see a red blood cell's too big? Okay? Now, I know it's hard to gather from this, but I'll also remind you, red blood cells have what inside of them? They have hemoglobin. Very good. And hemoglobin is what kind of molecule is it? It's protein. It's a big protein, actually, kind of, pretty good size. But we know 250 million of them fit inside of a red blood cell. Holy moly, don't remind us of that. We don't want to think about it. Well, you have a final exam coming up. It doesn't hurt you to think about it, right? But here's what we're saying, that even proteins are too big to get across this membrane. They stay in the blood and go through the efferent arteriole. Why? Here's the why, guys. Earlier in the semester, we talked about how in your blood you have plasma proteins. They're made to stay in the blood. If they could escape here, then you would lose most of your blood volume. You couldn't maintain your blood volume. Therefore, even here, those proteins cannot escape unless you have kidney damage, unless that glomerulus is torn. That's how we keep our blood volume. It's by allowing blood to come in. Almost everything gets filtered out. Not all of it, mind you, because we have to have blood cells and enough protein, and that keeps the concentration of water here and other chemicals as well. And then whatever does move across here is called glomerular what? Filtrate, enters the tubules. About 180 liters worth of water and other materials in there. And then we know that eventually 178 liters get pulled right back into the blood through a process called reabsorption. Okay? I do need you guys to listen to the lab, because I'm not giving you everything from the lab even now. I don't have time to re-lecture for all of that. So I've left off a lot of stuff, like the capillaries in here, fenestrated, and I go through all of that with you. I show you these little filtration slits and all that in the lab talk. Because what we have to talk about in here, how do we make the urine? Filtration, reabsorption, and secretion. Filtrate, tons a day, right? Reabsorption, tons a day. Secretion, not so much. Main things that are secreted? Medication, hydrogen ions, and potassium. And what would the hydrogen ions be regulating? pH. pH, right? So if we want the blood to be more acidic, we leave the hydrogen ions in the blood. But if we want to change the pH of the blood and let the blood to become a little more basic, we would secrete more hydrogen ions out. Also, we can mess with the amount of bicarbonate that leaves there that gets secreted. So that's the other major thing I should say. We said hydrogen ions and potassium and medication, and bicarbonate secretion can also help determine pH as well. It's kind of pretty neat, really. And what ends up leaving the tubes would be called urine. Should you have protein in your urine? I'm just asking you. Should there be protein in the urine normally? No. Should there be sugar in the urine normally? No. Should there be fat in the urine normally? The answer is also no. No, no, no. Okay? Those molecules are just too big, and or in the case of sugar, it's small enough to get in the filtrate. Look up there. Sugar gets filtered across here easily, ends up into the tubule. So what must happen to make sure that we don't have any in the urine? It gets immediately, all of it, reabsorbed right back into the blood. What determines if it moves into the blood? Take a wild guess. Maybe concentration? What do you think? High to low? Maybe? Just maybe? Now we'll talk about, there's some other things that go on here and we'll talk about that, but check this out. If you have too much sugar in your blood because you just ate a whole box of Twinkies at lunch, I don't think any of you would do that, so it's an exaggeration, I hope. But you know, Frappuccino is like Twinkie in liquid form, so maybe more of us have them in there than we think. If your blood sugar is too high, what happens is the sugar that ends up in the filtrate, in the filtrate, only so much can go back. You can only push so much back in if it's already full, right? That's why diabetes and people whose blood sugar gets sky high, diabetics have sugar in the urine because the concentration in the blood is just too high to get more sugar in. So it stays in the tubule. All of it cannot get reabsorbed. Once again, it's because of that concentration. Most of this is found in what part of a kidney? Well, this is a nephron. It's found in the cortex. That's where it's located. Sometimes, however, they have a big loop of Henle that digs down way deep into the medulla. Other times, it's just in the outer area of the medulla. This is happening to label cortex and medulla there, which is pretty good because it lets you see that. We will be discussing that again in more depth. Notice, they're showing water move here. They're showing solutes move here. I'll give you a little clue as to what I'm going to tell you next time. That's because solutes cannot move out of this tube. It's not permeable to them. So the concentration that's surrounding it sucks the water out. Then the concentration, it gets really concentrated. See where it was 300 up here? And now it's like 1,200 down here? It's because as we suck water out, it gets more and more concentrated. Do you guys understand that? Take water out, it makes the solute concentration greater. Now by the time it turns the corner here, this ascending loop of Henle, water cannot move out. So what happens? the solute gets sucked out by the water that's being pulled out here. Now the solute, or actually when they show it in red, they're telling you it's active transport and we're pushing it against the grain, is what they're telling you. So they pump it out, and then on this side more water comes out, and that allows finally the concentration here to change and go backwards. It's kind of technical. If you don't already own what filtration, reabsorption, and secretion are, you see this, and maybe it gets a little more difficult to understand. The easy part is, all the arrows are moving which way here? Out of the tubule. What do we call it when stuff moves out of the tubule? Reabsorption. This is a picture about reabsorption. They give it fancy names like countercurrent system or countercurrent multiplier. Why? Because it's moving down here. It's moving up here. But all along that, we focus on what moves out of the tubules, what's being reabsorbed in different areas.